We have been talking about cytoskeletal elements. We have said that there are three different types. Now, let me introduce you these different types of cytoskeletal elements individually. So, let me first of all tell you what are the three different types of cytoskeletal elements. There are intermediate filaments. They provide mechanical strength and resistance to shear and stress. Or we had said that there are two different types of forces. There are compressing forces and tensile forces. Intermediate filaments resist the tensile forces. Microtubules determine the position of membrane enclosed organelles and direct intracellular transport. I'm basically briefly telling the main points of these elements, cytoskeletal elements. There's, of course, a lot more to them than what is on the slide. We will talk about all of them gradually in different modules. Actin filaments determine the shape of the cell surface and are necessary for the whole cell locomotion as well. I hope you remember the slide I showed you when we were talking about cell junctions. Basically, the point here was that how the cytoskeleton is arranged in the cell and what are the different functions they perform. I had said that the plasma membrane does not have any structural strength. You can think of plasma membrane as a piece of cloth. If you put this piece of cloth on a ball, it will assume a spherical shape. If you put the same piece of cloth on a table, it will assume a square or rectangular shape. Plasma membrane is similar in that way. So whatever the structure is underneath the plasma membrane, it assumes that shape. So what are the structures underneath the plasma membrane that determine the shape of the cell? Let's talk about that. First of all, this bird cage I have shown in the slide, it has different components. First of all, let's talk about this hexagonal component shown here. This hexagonal mesh basically represents the spectrin. When we were watching a video, I showed you spectrin in that too. It is just beneath the plasma membrane, and it is supporting the plasma membrane and providing it a structural support. Underneath spectrin are the actin molecules. Actin molecules are basically, you can think of actin as the bars of this cage. They are basically facilitating not only the shape of spectrin, but also ultimately the plasma membrane. So the support is basically coming from actin, which is basically supporting the spectrin, and on top of spectrin, we have the plasma membrane. So the other structure you see here is the star. The star is attached to these hollow pipes. These hollow pipes basically are representing the microtubules. Microtubules themselves are hollow structures. We'll talk about microtubules in a different module. But these basically cytoskeletal elements, they basically are responsible for resisting the compressing forces. For example, compressing forces when you push on your skin. Additionally, you can see these brown strings or cords. These cords are representing the intermediate filaments. They transverse the cell. So these cords go from one side of the cell to the other side of cell. For the sake of simplicity, I had not made them in the other half. It doesn't mean that they don't exist there. So these elements, the intermediate filaments, they resist the tensile forces. Tensile forces for example, when I'm pulling on my skin. So this is basically a rough analogy of how cytoskeleton is organized inside a cell. Now let's look at them individually. Okay, we will do that in the next slide. But I would like to emphasize that usefulness of cytoskeleton in large part basically is a function of accessory proteins that link these elements. Without these accessory proteins, the cytoskeleton on their own cannot perform all the functions that they do. For example, we talked about how intermediate filaments or actin filaments are linked to adhesion molecules via these anchor proteins. So anchor proteins are also a type of protein, you can think of them as accessory proteins that basically modify the functions of cytoskeleton. These accessory proteins essential for controlled assembly or disassembly of cytoskeletal filaments in specific locations. Motor proteins, they are also accessory proteins of cytoskeleton. They can move organelles along the filaments, or they can move filaments relative to each other. Macromolecular components that make up these structures are in constant state of flux, rapidly responding to any eventuality. So just as we said, the ECM is not a static structure. Cytoskeleton is actually very dynamic structure. We will see how dynamic it is, and also it is adaptable, and it organizes the different components of the cell. You can think of cytoskeleton as, rather than as roads, you can think of them as ant trails. You have seen, if you place food somewhere, ants will form a nice line, and they will move towards the food. They will pick up little particles of food and move back them to the nest. So if you change the location of the food, the ants will change their basically ants will start going to a different location. So that is dynamic. They are not constantly going to the same location. They can change which location they go to. So the first cytoskeletal element I'm going to talk about is actin filaments. They're also referred to as microfilaments. They are basically two helical strands of polymers of G-actin. G basically means the globular actin. So there are two strands. You can see there is this strand and there is this other strand. They are forming this helix, and this is basically when these globular actins or actin subunits polymerize, they form this double helical structure. They are about 5 to 9 nanometer in diameter, and they form these linear bundles. Either they can form linear bundles of 2D network or 3D gels throughout the cell, but they're especially abundant beneath the plasma membrane. Remember the bars that were making the cage, so they're abundant just beneath the plasma membrane. They're supporting the plasma membrane, allowing a cell to form a specific shape. The next cytoskeletal element is the microtubules. They are hollow cylinders, and they're made up of tubulin subunits. We'll have a lot more to say about tubulin subunits later on, but I would like to point out that they have a 25 nanometer diameter, so they are rather much thicker than actin filaments. They are generally attached to a microtubule organizing center or centrosome, and almost all microtubules emanate from this microtubule organizing center in most cells. So the next category is the intermediate filaments. They are rope-like. They are about 10 nanometer in diameter. Intermediate filaments are polymers of subunits, which are actually themselves filaments. So you can see on your screen that this, for example, is one subunit of intermediate filament. So this is itself a filament on its own. They also are a very important component of the nuclear membrane. They form these nuclear lamina. And these cytoskeletal elements, besides these functions, they perform a lot of other functions, which we will talk about in later modules.